Hi, this is the AP Bio lecture on lipids, and your essential question is, what are lipids composed of and what do they do in the cell? You should be able to answer this question with all of these vocabulary by the end of this lecture. Let's start with the characteristics of lipids, and then we'll get into some important types of lipids. So first of all, remember that if at any time you need to pause the video to read through the slides, or if you need to leave the video to get a firmer understanding of the concepts underlying this stuff before you come back to it, you're more than welcome to do so. And these slides are in our Google Drive folder as well. The defining characteristic of lipids is the presence of hydrocarbons. So a hydrocarbon is just any molecule that is mostly carbons and hydrogens, and that's pretty much it. Now you'll see with this fatty acid, you do have a carboxyl group here, the C double bond O OH, but the rest of it is just carbon and hydrogen. Now you have this glycerol molecule over here, and it's got 3 hydroxyls here, here, and here. And remember that a hydroxyl can interact with things like a carboxyl in this case. And what they do is they form a dehydration synthesis reaction to form an ester bond. And so there's going to be, as it loses this water molecule here, that's going to cause a new bond to form between this oxygen here and this carbon here. It's what we call an ester linkage bond. And so that is going to link this fatty acid to this glycerol molecule. And because the glycerol molecule has 3 total hydroxyls, it can actually form ester linkages with 3 fatty acid chains. And when that happens, we call this a triacyl glycerol or a triglyceride. When we're talking about fats, the triglyceride is essentially kind of the monomer of a fat. It's not a true monomer because fat molecules, they don't really bond with each other to form really long, big biomolecules, but they do associate with each other through what we call hydrophobic interactions, which means that because they all hate water, they're going to clump together so that they can avoid water as much as possible. And that's the equivalent of a large biomolecule, even though it's not a true bond. And so that's what that essentially says here. Fats separate from water, and they clump together through what we call hydrophobic interactions. So here are some examples of fats. You can see that this is a saturated fat, and the reason is it's got 3 fully saturated fatty acid chains, where in this model, the carbon is represented by the black triangles, oxygen is in red, and then all of the hydrogens are in gray. And you can see that there is the maximum possible hydrogens that you can get because you only have single bonds among all of the molecules here, or all of the atoms here. Here's another representation of that. So around every carbon, you have the maximum number of hydrogens here and here, and then around this final carbon, you have 3 hydrogens. So we say that this is a fully saturated fatty acid chain. They are completely straight. In comparison, this is an unsaturated fatty acid chain. And the reason why is there's a double bond right here between the carbon here and the carbon here. And that's going to cause the molecule to bend. It's going to cause it to kink because of that double bond. And it's going to prevent you from having a full saturation of hydrogens. So you're going to have less than the maximum number of hydrogens that you can get. Here's another thing you need to know about saturated and unsaturated fats. When you have completely straight fatty acid chains, they can pack together really, really tightly, and so they actually form these solids. An example of that is butter. But when you have an unsaturated fatty acid, even if it's only 1 chain out of the 3 that's bent, because of that bend, it prevents these triglycerides from packing together tightly, so they actually stay fluid at room temperature. An example of that is olive oil. Now, it's not just fats that can bend and that have solidity and fluidity depending on the number of bonds that are there, double bonds that are there. We're also going to see that in phospholipids as well. Before we get to phospholipids, just to wrap this up, you should know that the major function of fats is energy storage, and that's particularly important for animals, multicellular animals, because fat is a way for animals to store energy and carry their energy with them so that even if they're not able to eat for long periods of time, they can live off of the energy that's found in their fat. And the reason why fat has so much energy is, once again, those hydrocarbons right there. There's so many bonds that can be used to basically release energy, because as these bonds break, they'll form new substances, and as they form new bonds and make new substances, they'll release lots and lots and lots of energy for the animal to use. Plants also obviously make fats. That's why olives are used to make olive oil. Okay, next. Another type of lipid is the phospholipid. And so this is not going to be an actual fat because it's not made of triglycerides. But the reason why it's not a full triglyceride is because it's actually really just kind of a diglyceride, where here's your glycerol right here. This was your glycerol. Through dehydration synthesis, it formed ester linkages with two hydrocarbons, two fatty acids. So here's one straight one, and then here's one bent one. But then that third hydroxyl on the glycerol actually did not bond with another fatty acid chain. Instead, it bonded with this structure here, and the defining characteristic of this structure is the presence of the phosphate group right there. You'll notice about the phosphate group that it has this negative charge right here, which means that it is hydrophilic, because remember that water is attracted to negatively charged particles. On the other hand, the phospholipid also has these fatty acid tails, which, because they're hydrocarbons, they're hydrophobic. And so a phospholipid has a phosphate head, which is hydrophilic, and fatty acid tails, which are hydrophobic. Here is the ball and space-filled model. And then here is a simple representation. And as you must remember from 9th grade, phospholipids form a double layer in water. So there's water outside, there's water inside, and the fatty acid tails, because they're hydrophobic, they get sandwiched in the middle. Next, let's talk about steroids. So steroids are lipids that are also hydrocarbons, but the difference here is that the hydrocarbons are fused into ring structures. I want you to remember that when you see a structure like this, every vertex has a carbon, and it has like usually a couple of invisible hydrogens. Here we have a carbon, here we have a carbon, this is a double bond. And then you can see there's a methyl group sticking out here, there's a hydroxyl group sticking out here, here's another methyl group, and then over here we have a hydrocarbon chain. So here's a carbon, here's a carbon. Notice there are no oxygens anywhere in this structure except right here at this one hydroxyl. So when you see 4 fused rings like this, with some other stuff on there, and everything is just hydrogen and carbon except for one hydroxyl, then you know you're looking at cholesterol. And cholesterol is a significant steroid, where a steroid is any lipid that has this fused ring structure. Cholesterol is a significant steroid for a few reasons. First of all, cholesterol is a precursor to estrogen and testosterone. And so cholesterol can be used to make those steroid hormones that then go throughout your entire body and signal your body to do different things like, grow hair or grow muscle or stuff like that, make a baby. But in addition to that, cholesterol in its natural form is also a really important component of cell membranes. So you know that high cholesterol is bad because it can lead to cardiovascular disease, but cholesterol is an essential component because it's part of your cell membranes. So here we're going to look at your cell membrane under a few different conditions. One is if your cell membrane is too fluid. Why would that be? Well, you'll notice that the phospholipids are not able to pack tightly together because look at these little bent chains over here, just like kicking out and preventing other phospholipids from getting close enough. When the phospholipids are too fluid, the cell membrane will fall apart, and that cell is not going to be stable. Over here, the phospholipids are too viscous, so they have only saturated fatty acid chains. They're packed too tightly together, and they're not fluid enough to allow substances to, cross in and out naturally. And so this cell is also not going to make it. But if you have a mix of saturated and unsaturated fatty acids in your phospholipids, then you're going to have a fluid membrane that is still stable. And it's going to be even more stable if you have these cholesterol molecules. So you can see the 4 fused rings in here. These cholesterol molecules interspersed among the phospholipids do a couple of things. The first is at moderate temperatures, they actually provide structure, like they kind of like anchor the phospholipids so that they provide stability, so that they keep the membrane from becoming too fluid. But then at low temperatures, as the phospholipids slow down and they start packing tightly together, cholesterol actually spaces them apart and prevents them from packing too tightly, so that the cell membrane is prevented from solidifying, and it maintains some fluidity of structure, which then allows it to maintain its function. And here's all of that in words. If you want to pause it, you can, so you can read the bullets. Okay, here are your key takeaways. Lipids are hydrophobic, which means they're excluded from hydrogen bonding with water, so they don't mix with water. The reason for that is they're composed of hydrocarbons that are bonded with nonpolar covalent bonds, so they cannot mix with water. There are a few different types of lipids. There's fats, phospholipids, and steroids. We're not going to talk about waxes in this lecture, but those are out there too. So fats store energy. If you have saturated fatty acids, they have straight chains. They pack together solidly at room temperature. And if you have unsaturated fatty acids, they have at least one bent chain, and they're fluid at room temperature. If you have phospholipids, then instead of having a triglyceride, you're going to have 2 fatty acid chains plus a phosphate. And those are great for forming cell membranes. So the hydrophobic fatty acid chains will form the interior of the membrane. And the more unsaturated fatty acid chains you have, the more fluid the membrane is going to be. And then last of all, you have steroids, which serve as signaling molecules throughout the body. So think of estrogen, think of testosterone. And in addition to that, you have the steroid cholesterol, which stabilizes cell membranes. I'd like you guys to go back to the essential question and pause the video and see if you can answer this question using all of these vocabulary. If you can, that means that you have mastered this topic.